Medicine, and it's my pleasure to introduce our Otto Schmitt distinguished lecturer today. So I'll start by just telling you a little bit about what the lectureship is. It's designed to foster a discussion of the grand challenges for engineering and medicine, and we invite national opinion leaders in research, industry, and government to give 1 or 2 talks each academic year. And these public lectures are named in honor of Dr. Otto Schmitt, a longtime university professor of biophysics, bioengineering, and electrical engineering here at the University of Minnesota. And he's known for his scientific contributions, but also for establishing the field of biomedical engineering. And so, without further ado, I will introduce our lecturer today, Dr. Lori Setton. Dr. Setton received her bachelor's from Princeton University in mechanical and aerospace engineering, and then her master's and PhDs from Columbia. She joined the Department of Biomedical Engineering and Orthopedic Surgery at Duke in 1994, and she rose up through the ranks, becoming a distinguished professor of biomedical engineering and orthopedic surgery there. She then more recently moved to Washington University to accept the position as the Lopata Distinguished Professor of Biomedical Engineering and Orthopedic Surgery there, and also to become the chair of the Department of Biomedical Engineering. Her research focuses on understanding the mechanisms for degeneration and regeneration of soft tissues of the musculoskeletal system. And for this work, she's been funded through a variety of different foundations and federal agencies, NIH, NSF, Whitaker, Coulter Foundation, and many others. She has over 170 peer-reviewed articles. Her IP has been licensed by several companies for commercial development. She's been on the editorial boards of multiple journals that are leading journals in our field: Annual Reviews of Biomedical Engineering, Journal of Biomechanical Engineering, Osteoarthritis and Cartilage, and the Journal of Biomechanics. She's also served on the boards of Orthopedic Research Society, the World Congress of Biomechanics, and most recently the president of the Biomedical Engineering Society. She's a fellow of many of these societies: ASME, BMES, as well as AIMBE, and she's also a winner of the ASME Mal Award, and she actually, I think, presided over that award the year when I also won that award. So it's with it's an honor and a privilege to have Dr. Setton with us. Let's welcome her to the stage. Thank you.
Thank you so much for that very generous and lengthy introduction. So when I was invited to come give this lecture here, at least 2 faculty wrote to me with long paragraphs and statements. Did I know who Otto Schmitt was? And I am deeply offended. I'm going to see who knows more about Otto Schmitt, me or you. So this is a set of slides that I have never had. I didn't make special for this. I've made these before. So show me by a hand, show me your hands. How many of you know where Otto Schmitt was born? Aha. St. Louis. He was actually born in St. Louis in a region that was a very historically black region, and is very close now to an area that you have heard of, close to Ferguson, Florissant, that area in St. Louis. So next question, let's see if we can figure this one out. Where did he go to college? Washington University. That's right. And why did he go there? This is who we celebrate. We celebrate, and his face is on our wall, Francis Otto Schmitt, who's 10 years older than Otto himself. And Francis was already on the faculty at WashU when Otto was in high school. So Otto did his first experiments in Francis's lab. And that's what made him such a prodigy, so that he graduated quite early. I think before he was 18 years old, he got his bachelor's degree. So then he stayed on and he got his Ph.D. And remember, his brother was in this area that eventually became physiology. And that's how Otto took his interest and training in physics to start to study impulses along nerve fibers, which led into the Schmitt trigger. Very, very famous Schmitt trigger. Of course, you know him and celebrate him here because he came and he served on your faculty, I think from like 1947 to 1983. So no, it must have been earlier than that, 1942 to 1983. And why did I have to learn all of this? You'll see in a minute. So Otto was a big shaper of our community and the community that we enjoy today. And he's responsible for forming a very important society that's known to us. Does anyone know the name of this society? You're right and you're wrong. What was the first society that Otto Schmitt formed? This should be easy because this is like an obvious. I can't see. I told you. I joined the faculty at WashU 4 years ago, and I was trained in this. So he formed the Biophysical Society, and he did that in 1958, and he set up the bylaws and the charters for that society that eventually became the Biomedical Engineering Society, as you'll see. And it was really this guy who was at the NIH who was bringing together people like Otto Schmitt, electrical engineers who were interested in biomedical problems. But it was Otto who stood up to become the first president. They had a budget of $2,000 when they got started. And why did I have to learn all of this? Because I was elected to become president of BMES in 2016, and I didn't pay attention to the tiny print that said that the society would celebrate its 50th anniversary in 2018. So I had to run the 2018 meeting when we were celebrating the 50th. So suddenly I had to write all this text, and I actually had to go to the museum archives and go through files and files to take photos to put together a timeline that we used. And here's Otto at the 25th. And the presidents, the living presidents at the time of the 25th took this photo, and you can see Otto is there all the way on your right. And this is actually Jack Brown. So they were still very active and involved in the society. You'll recognize some others probably. Peter Katona, Y.C. Feng. And here we are in the 50th. So we tried to reproduce and recreate that staging. These are all the living presidents that we could get together. And this is our honoring the Whitaker Foundation, that's Jack Linehan, for their support of BME and the BMES. So if you learned nothing else today, you learned about Otto Schmitt. All right, but you asked me to come here to talk about research, and you heard that I was a classically trained mechanical engineer. And so at the beginning of my career, I was involved in looking at micromechanics of tissues and then eventually of cells and looking at cellular systems. That led us to begin to ask questions, and I'll talk a little bit about it today, about specific cellular phenotypes and stem cells for differentiation and regeneration. Our group is very active in engineering biomaterials that can be bioinstructive and inductive, and we measure functional success, meaning outcomes of the procedures that we assess and develop. And as part of our therapeutic strategy, we focus on materials, but also on drug depots for delivering known inflammatory antagonists. And our work is focused historically quite a bit on arthritis, but today I'm going to talk to you about the spine and the intervertebral disc, because I think in the spirit of the Otto Schmitt lecture, this is one of the grand challenges. We have a pain epidemic in America, which became a opioid epidemic, and there's no question that back and neck pain are the largest contributors to disability in this context. And we still really do not know enough to be able to be successful as biomedical engineers in developing interventions and in attenuating any pathology that occurs. So all I'm showing you here is every 10 years we collect the state of the U.S. health, and we're about to do it again. But back when this was published in 2013, low back pain and neck pain were the #1 and #4 greatest contributors to disease burden in the U.S. And what's important about that is that intervertebral disc disorders contribute to a significant percentage of that, well over 40% and much higher in the neck. So I'm going to get everything you need to know about this structure that you probably don't think about a lot in this one slide. So the intervertebral disc is a spacer, obviously shown here between the vertebral bodies in your spine, and it's responsible for flexibility and motion. So if you're a mechanical engineer like I was, right, you're going to find this a very interesting structure because it's clearly subjected to, compression, tension, torsion. It's very interesting in how it achieves its goals, it achieves its purpose. And it does this largely through generation of an internal pressurization that occurs due to the presence of these negatively charged proteoglycans that contribute to a very large swelling of the structure. And that swelling is distributed isotropically, if you will, equally in all directions, to a restraining layer that we call the anulus fibrosus. And that's probably all you're going to need to know about that structure for what I'm going to talk about today, because we're going to very quickly see that mechanics is not necessarily the major question. So I mentioned, or maybe John mentioned, that before I moved to WashU, I was at Duke, and I had been studying these problems for a decade. But when I moved to the Midwest, this was the question I was asked. No one really cared about, what are the mechanisms for degeneration in the intervertebral disc and how does this happen? But they all asked me, what happened to Peyton Manning? So I wasn't, coming from North Carolina, I wasn't terribly familiar with Peyton Manning, but now I can tell you what happened to Peyton Manning. How many of you have heard of Peyton Manning? Look at that. I'm in the Midwest. I think so. He had a series of debilitating disc disorders. And this is a disc from like a 20-year-old, and this is a disc that really could come from like a 40-year-old. And so what you're seeing, and this is a natural process of aging, what you're seeing is this loss of hydration. So that structure in the middle that's responsible for internal pressurization, called the nucleus pulposus, right, that undergoes this fibrosis, this fibrotic stiffening. And what that means is that there are many different manifestations of this kind of change that can occur. You can have a loss of disc height, which has reduced motion, and that's what you're seeing here. You can also have a situation in which you have an actual extrusion of the disc material from the anatomic compartment. So the disc is avascular, aneural, and alymphatic, and it's supposed to stay contained within that annulus fibrosus, but it can actually displace anatomically onto adjacent structures. And you have nerve roots to the left and right, and the spinal cord posteriorly. Anteriorly, you have the aortic, the abdominal aorta. So it's really kind of important that it stay anatomically in its setting. And when that happens to you in your neck, you have tremendous pain and disability that can occur. And in the case of Peyton Manning, this is just a diagram showing you all the different ways in which those anatomic changes can contribute to pathology. He wound up having a series of 4 surgical procedures until the only thing that actually worked for him was to have a plate placed on his spine, and you can sort of see this sort of grafting and plating here, and that allowed him to obviously be traded to the Broncos and then go on to win the Super Bowl. So it's a good story, and we like to think that we know how to handle spinal pathologies and interventions, but this is an incredibly, as you can see, 4 procedures, an incredibly disabling surgical intervention. And what we learn about this is that the only way to stop the pain is to stop the motion. And that's true of a fractured bone, for example. So we know that there's some connection between motion and all the negative consequences of that and the resulting symptoms of pain. But what we have come to understand and learn is that it's far more complex than that simple explanation. So when a disc is undergoing that fibrotic changes that I just showed you in that diagram, there are a series of cytokines that will be secreted by infiltrating cells. So cells that don't belong there will start to produce protein, usually protein-based molecules that will drive an inflammation or also promote a nerve infiltration and a series of pathological changes such as activation of the microglia in those adjacent nerves that will drive a neuropathology that is induced completely by crosstalk between that musculoskeletal tissue and the adjacent nerves. And what I'll tell you a story about our first therapeutic intervention in this case, just one of several strategies that we tried. We knew in inflammation and musculoskeletal diseases, and as we know in many others, this is now back in the early 2000s, that one of those molecules that was being produced in the disc was tumor necrosis factor, a very potent pro-inflammatory cytokine. And at the same time, a series of protein drugs had been made recombinantly by major pharma companies. For many of them, this was their first foray into biologics. They were synthesizing blockers, so basically receptor blockers that would bind to the TNF and inhibit the signaling. So you'll see we refer to these as TNF receptors, but they're soluble TNF receptors, so they're not membrane bound. And they were very, very effective and are still used today clinically. These are blockbuster drugs used in the treatment of rheumatoid arthritis, inflammatory bowel disease, Crohn's disease, and more. So it was very logical in the early 2000s that TNF should be attenuated or blocked in the treatment of disc-related pathology. And so there were two major clinical studies, one out of Hopkins, administering the drug via epidural administration or also by intravenous administration. And in both cases, they failed clinical trials. And they failed to reduce the pain that was associated with the discogenic pathology. And this is perfect for an engineer. Like myself, I was very interested in local drug delivery, and we had been injecting drugs in knee joints for a while. And we basically said for focal pathology like discogenic pathology that systemic treatment, so any kind of intravenous or epidural treating the entire host essentially was not indicated for a focal pathology like low back pain. And you would never want to do this with a big, with a protein drug like this because it's a big immunosuppressant. So you really don't need, for the case of it's just like a herniated disc or just degenerative disc disease, you do not want to frequently administer these drugs, and they're very, very expensive. So we knew this was not a therapeutic avenue that we were interested in. So at the time, I was at Duke, and I had a student who was friends with a student in the lab next door, which was Tosh Chilcote's lab. And they had been very interested in recombinant protein expression of this polypeptide called elastin-like polypeptide. It's a series of pentapeptide repeats that undergoes a hydrophobic self-association, and you just sort of saw that there in that cartoon, in this little movie. It's soluble at, say, room temperature, but it'll undergo a hydrophobic self-association to form multimeric particles, and that's what you're seeing here. So this is a elastin-like polypeptide tail surrounded by water molecules, but if you raise the temperature above an engineered design temperature, it'll shed its water molecules and it'll form particles that are of the order of 1 micron or so. And so we reasoned that since these biologic drugs were being made recombinantly, and this elastin-like polypeptide could be made recombinantly, that we would subclone the gene for the human soluble TNF receptor onto the genes for these polypeptides, and we would be able to make a class of drugs that would have an ability to be sequestered from the site of delivery and then slowly released over time, and I'll show you that here. I'm showing you that we picked one fusion protein. It was one of, I think we made like 12 of them to figure out which one works, so I saved a few slides there. But we picked a protein that would transition, so form those multimeric particles at 32 degrees. So you could inject it into the pathology site, which in our case we were focusing on the nerve root or the dorsal root ganglia. And the surgeon liked it because it was clear in the syringe, and he or she was doing what they always do, which is just inject, do like a nerve block, and they were injecting it just at that site, and there was no change to their actual procedures, but the nerve would get covered with these protein deposits, and the idea is that when those protein deposits, which I have aggregates here, were at that site, they would undergo this non-equilibrium thermodynamics on and off from the aggregate, be available to bind to the drug or block the receptor, because we did this with some other drugs as well, and then be cleared from the site in their freely soluble form. So when we did this, we found a couple of things that were of great interest to people interested in translation. We found that we had a 7-fold increase in the half-life, so we could keep a drug at this site 7 times longer than if we just injected the protein drug by itself. When we do this in the knee joint, we get a 25-fold increase in the half-life. So it's just really been huge, and it depends on the site that you choose for injection. But importantly, because remember I said that these were potent immunosuppressants that we were working with, we were able to not have all the drug that you deliver get dumped into the systemic circulation all at once. We were able to drop back the peak serum exposure to the drug by 10-fold. So we needed to demonstrate proof of concept, right, that this is sort of the preclinical translational pathway. And so what we did was we actually induced an inflammation by placing the nucleus pulposus tissue on a dorsal root ganglia, and we did this in a rat, and when you do that, what you're seeing here is just like what you see in a human. And so what you want to look at here is this NP. This is nucleus pulposus tissue alone. And this is showing the percentage of time spent on 1 foot. So humans do this as well when they're in pain. There's a couple of things they do. They move less. So you'll see me talk a little bit about freely selected behaviors. They have lesser freely selected activity. But they also will not weight bear on the affected limb. And so we found through a series of gait studies that they had reduced stance time, changes in stride length in this model. And this is an axial force transducer that you're seeing here. And so they had a lowered peak axial force for that limb for the side that was affected. And Kyle Allen went on to become a professor at University of Florida. Some of you might know him. And when we were able to locally deliver the drug at that perineural site, we were able to restore, and this is now only at 1 week, but we were able to restore the normal ambulation and the normal pattern of walking and the normal weight bearing. So here, this is the blue, the normal axial force weight bearing on that limb, telling us that if we were able to block inflammation at the site of pain transmission, that we would be able to at least restore function. And that, in many of the cases, that's what we're all interested in doing. And there were some other things that bothered us about this. You heard me mention that I said we looked only at 1 week, and we would consider that an acute pain presentation. We found a lot of other things that happened in this model, and TNF was our target, what we thought was our quote-unquote pivotal cytokine, but there were a ton of other cytokines that were involved, like interleukin-6 and interleukin-17. And we knew that these were, even though we might be able to take away the pain, we knew that the pathology was persisting, and that if we didn't keep the drug there for longer, we would be running into some trouble. So we became more focused on chronic pain. And what we knew was that if you have a pathology down here in the lumbar region, so that's the L5, L6 that I'm showing right there, and that would be, let's say, the most common site of discogenic pathology in humans. We knew that the major, and this is from a series of neuronal mapping studies that came mostly out of Japan, we knew that most of the innervation of this segment was through the sympathetic ganglion, and it passed through 3 dorsal root ganglia that were superior to the site of the injury. And of course, this is true for like the knee or the ankle or the hip or anything, right? They are all mapping to roots that are responsible for transmitting the pain to the brain so that you experience your symptoms as pain. So we became very interested in the idea that we could treat, not down here, the site of pathology, but that we should be able to treat the path for pain transmission in this pathology or in any other pathology at a site distal to the actual site of the injury. So we started looking at, and this is more recent work, so just to kind of show you the path that we took in a project like this. So we, in this case, punctured that lower lumbar disc. And remember, we think that the pain is being transmitted through dorsal root ganglia that are several segments away. And in this particular study, we were interested, the red here is supposed to denote nerve growth factor. And the nerve growth factor is like tumor necrosis factor in that it's secreted in discs. It shouldn't be there normally, but it's there when there's a substantive amount of pathology. And that nerve growth factor, Liz hypothesized, so a Ph.D. student who just finished earlier this year, Liz hypothesized that this nerve growth factor would actually bind to receptors. There's a track A and track B, and P75NTR are receptors on dorsal root ganglions that are mediated by binding to nerve growth factor. And she hypothesized that this was a mechanism that induced, was responsible for an elevated sensitivity to pain in this model. And other groups had already documented, as did we, that there were changes in the animal gait. And this is just showing you an example out at 20 weeks. So that's what we're calling chronic injury now. Out at 20 weeks, there's substantive changes to the anatomy and morphology of the disc that's consistent with what we would see in humans. So Liz studied their behavior, and in this case, we were doing freely selected behaviors. So she started to see a pattern of reduced time traveling. So the animals were selecting to be less active, starting at about 10 weeks or so. And then she also showed changes in gait, working here with an undergraduate at WashU. She started to show changes in gait out at these later times, suggestive of a pattern of an onset of injury with chronic pathology. All right, so remember she was interested in NGF. And so NGF here is shown binding to a cell membrane on a cartoon of a neuron. And And one of the major ways in which NGF is believed to mediate pain in neurons is by regulating the insertion of a trip channel in the cell membrane. So the trip channels can be internalized, or they can actually be inserted in the membrane, where they can be active in transmitting mostly heat. So this is the capsaicin receptor, so mostly heat through the trip channels. So we're going to look at trip channel activity in neurons at that chronic time point of following intervertebral disc injury. And so what Liz did was isolate neurons and expose them to capsaicin to test the activity of those trip channels, which are involved in transmitting heat and pain, and then looked at NGF to see if there is a sensitization to NGF in these neurons as she had hypothesized. And this is just showing you in this initial work, we were working with calcium-sensitive dyes to look at this as a measure of surrogate activity for trip channel activation. And this is a lot of data, but you just follow what I'm circling here. The black is the discs that were punctured, right? So those are the injured segments. And these are the dorsal root ganglion neurons from the injured segments. And what you can see is that in all cases, we had fewer neurons responding to trip activation and fewer trip-positive neurons in these cells. So we definitely saw evidence of remodeling, but we were a little bit surprised. We thought we would see elevated expression of trip channel in neurons that were involved in more in mediating pain. But in fact, we did not see that, which suggests that maybe some of the changes involved in internalization of that trip channel, which left us knowing that there are important changes and there are going to be druggable targets, but that we hadn't yet found those druggable targets. So what we're doing now is actually just finished setting up a system using transgenic mice that are labeled with GCAMP, GFP signal. For some of you who might be familiar with that, it is a transgene for a protein that is sensitive to calcium stimulus. And so these animals are expressing this in the dorsal root ganglion. And we can image, not getting my movie here, but we can image dorsal root ganglia in vivo to look at the remodeling responses that we might see in response to chronic disc injury. And in that setting, we can look at druggable targets like specific sodium channels to see if we can identify the correct target there. Probably the most important thing that came out of so much of this work is that the NIH has pulled together not only a roundtable and issued some more funding announcements to accelerate research on the intervertebral disc, because what we've really revealed is how little we know about how we can see degeneration translate into pain. And then that was back in 2016. In 2018, of course, with the opioid epidemic now, the NIH has partnered with other entities, so it's like a trans institute initiative called the HEAL Initiative, which is to tackle all contributors to pain in the global population. And so DISC is definitely an area that is continuing to be targeted, and of course very much needing additional people to work in this area. So I wanted to have a translational story, and a story that told you a little bit about one of the sort of global challenges that we face as bioengineers trying to develop therapies for human pathology. But the title of my talk originally was, the death and rebirth of the intervertebral disc cell. And that story is also one that we're very passionate about and has also informed translational strategies. So I'll talk a little bit about that. So that's my sort of cellular therapy story. But you know, you saw the cartoon, and you can see it again here, of the changes that occur in the disc with aging, and almost all of these changes can be attributed to a death of the cell population that occurs in this region. If the cells could stay alive, they would make proteoglycans, the disc would maintain its swelling pressure, it would actually contribute to load transfer to the anulus fibrosus, and it could continue to function throughout the life of an individual. So why do these cells die? So this is one of the largest avascular structures in the body. There's no oxygen, there's very low glucose, there's very high pH, low pH, I'm sorry. And there's also potential mechanical involvement. With this cell death, what happens, we get this cycle. As cells die, we lose that pressurization, we see elevated matrix strains, and we think that that is sort of this vicious cycle that continues. And as the cell starts to, as the disc starts to fracture, we can start to see other cell populations move in, like macrophages and lymphocytes, and T cells, and those will actually drive a proteolytic enzymatic damage. So a lot of interest in why these cells are not optimized to survive in this environment. And so I'm going to take you a little bit through this unique cell type. So those cells in the nucleus pulposus are derived from notochord, and the notochord also contributes to the formation of the spinal cord. And one of the sort of distinct morphological features of these notochordal cells is that they are highly vacuolated, and you'll see a picture of that right here. So this is a NC stands for notochord, and they have these big sort of blebbed bodies in them that are supposed to be there during growth and development. And we believe that they, the mechanical inflation of those vacuoles are necessary to contribute to the turgidity of the notochord during growth and development. And they stick around in the human, in the postnatal human, for about anywhere from 3 to 7 years. And they continue to make extracellular matrix, and they more than double their tenfold increase in their tissue volume over a period of 3 years. So we know they're very biosynthetically active in the juvenile form. So for I think almost all of my education, I was told these cells are like chondrocytes. And chondrocytes are derived from mesenchymal cells, and so that's the mesenchymal cells in chondrogenesis. That's what I refer to here. And when you're talking about chondrocytes, you care about collagen and aggrecan, or proteoglycan, and you're very interested in TGF-beta and Wnt signaling, for example. And some of those things are very relevant in this cell population. But there are very important distinct differences that I'm going to be talking about in the talk, and some of them have to do with being optimized to be in a low oxygen environment. So when cancer biologists started to move into this field, we started to learn a lot about molecules like HIF-1 alpha, which is very important, glucose transporter, brachyuria, an important transcription factor in some cancers, but also an important transcription factor for controlling morphogenesis in the notochord. So they are distinct, and so that's great because we can separate these two cell populations. And at the beginning, I was very interested in doing stem cell differentiation to get these cells back. And we really have been under a lot of pressure to demonstrate that we can get re-expression of these important molecules. But what I want to tell you about today is that sort of when we were looking at these cells and just looking at histology, we saw that they lived, the nucleus pulposus cells lived in these little colonies. And you don't see that in the anulus fibrosus cells or in tendon cells or really even in cells of ligaments and other highly collagenous tissues. And so I had a Ph.D. student who I said, well, just to, remember I'm an engineer and I'm a mechanical engineer, so I was like, help me understand what the mechanical environment is for a cell that lives in one of these anatomically fidelic environments versus a cell that might be sitting by itself in its own pericellular matrix. And so she did a lot of finite element modeling, and a lot of times I give talks all about that. But what I'm going to do now is tell you about the images that she brought to me because she had to identify this pericellular matrix region. And she came to me and said, the only way I can distinguish this nucleus pulposus region from the anulus fibrosus region is through the presence of multiple forms of laminin. And no one who works in cartilage or chondrocytes talks about laminin. Laminin is not there. And so we were pretty puzzled by this until a developmental biologist kind of laughed at us and said, well, laminin is a really important component of the notochordal sheath, so duh, of course it's there. And we didn't really pay too much attention to that. But then being interested in sort of cell mechanics, what we did was, this is a cell attachment assay, and we found that these cells preferred to attach to any isoform of laminin over fibronectin or collagen. And we spent a lot of years looking at integrin subunits that mediated the interaction with laminin, and basically it's like alpha-3 and alpha-6. Beta-1 integrins are really important in mediating those interactions. But then, this was after like maybe 15 years of the community being interested in the role of stiffness in mediating cell differentiation. And we knew, well, we had like the best tissue for studying stiffness because we knew that it was highly gelatinous when it was juvenile, but that the stiffness would increase dramatically and become fibrotic with aging. So we did what a number of people did, which was sort of reproduce the experiments of like Chris Chen and Adam Engler and Dennis Disher, where we worked with a polyacrylamide system that we could control the stiffness, and then we would pattern different extracellular matrix molecules on top. And so we have different stiffnesses that you'll see here, and BME is basically Matrigel, which is a laminin-rich mix. And what you'll see is we go from soft to stiff, we see big differences in the morphology. And we couldn't reproduce that with type 1 or type 2 collagen, as I'm showing here. And these slides don't have the contractility, but we also did alpha smooth muscle actin. And you can see that on the stiff substrates, we can reproduce a contractility. But when we put these cells, and this is Priscilla Huang, who's now has already left and has joined the faculty at Virginia Commonwealth, Department of Biomedical Engineering. These cells, when you put them down on a soft substrate, they just moved around until they found each other, and then they formed these colonies. And when they formed these colonies, they had the highest, so this is going to be the leftmost panel here, and I'll show you with the arrow. They had the highest production of proteoglycan, and they had the highest expression of some of the most important markers, brachyury, type 2 collagen, and N-cadherin, or CDH2. So we felt that by reproducing that morphology and using engineered substrates to do that, we could control the cellular fate and the cellular biosynthesis. And Priscilla actually did her whole thesis on cadherins and looking at cadherin signaling. But in the interest of thinking about, how we looked at this for translation, starting in the 90s, there were companies that were very eager to take cells out of your body, expand them, and inject them back in. And for the disc, the leader in this is a German company called Codon. But there's quite a few others. Mesoblast is probably the largest one now, working with allogeneic cells. But there are a few others. And a lot of interest in cell sources that you can inject back into the site. And so we looked at this and we said, well, they get the cells in, but there are a ton of FDA concerns about where the cells go and what they do when they leave the site, because if you have to inject them through a hole, they come out through the hole. And we were very interested, therefore, in developing biomaterials that could serve as cell carriers. And so our logic, of course, is that you need to present the laminin in order to get the cell to be the right kind of cell. So we made a host of engineered laminin-presenting gels, and that's what you're seeing here. These are pegylated, so we basically put polyethylene glycol. We decorated laminin to the point where we had cross-linkable moieties, but we didn't dramatically modify function. And this is showing UV light cross-linking, but we also work with a thiol acrylate and maleimide acrylate formulations that we can inject. And we can control the stiffness, and all you need to see here is that we can reproduce those behaviors in 3D that I showed you with Priscilla's work. And then we can inject back in, and we can show that here we weren't looking at a very long period of time, but we can show that if you just inject the cells in PBS, they're gone. But if you inject them in this PEG laminin hydrogel, we can keep them in that site for an extended period of time. So I know I'm at like 12:38. Does this go to 12:40? 12:50? So you want a more basic science story? Or I can take questions. But you like basic science? Okay. So obviously we wanted to know, how is the cellular interaction with the laminin? How is the cellular interaction with cadherin, which I'm not going to talk about? How is this controlling the state of differentiation of these cells? And so when you talk about fibrosis, we've learned a lot about the role of two promoter elements, TEAD and SRF. And we had learned about them from cardiomyocytes at sites of infarction where there's a stiffening. These were believed to be activated. And when they are activated, they drive genes associated with fibrotic phenotypes. And so Bailey, who is now assistant research professor at Carolina's Medical Center, she demonstrated that when you're on a stiff substrate, integrins cluster, and F-actin fibrils form. And when that occurs, the MRDIF, which is a cofactor driving SRF, or YAP-TAZ, which is a cofactor driving TEAD, can actually translocate. They are free to translocate to the nucleus and drive this expression. But on soft substrates, and she did this animation, so I'm not quite sure what it's doing. I think something's going to fly, yeah. Right, so when you have a soft substrate and your integrins don't cluster, your F-actin gets converted to G-actin, and it's available to bind to these cofactors, MRDIF, and it will phosphorylate YAP-TAZ to prevent it from translocating to the nucleus. So remember, this is her hypothesis about how soft substrates inhibit fibrotic differentiation of these cells. And so she used the PEG laminin system and two different stiffnesses, and she cultured human cells and reproduced. This is all just reproducing the biosynthesis that Priscilla had seen, and this is the light green here is showing you the higher expression of aggrecan and proteoglycan and that more rounded morphology. But when she looked at the activation, remember, of those promoter elements, so here SRF bound when MRDIF binds to SRF, she found, as she had proposed, that there was an attenuation of that fibrotic signal through that element SRF. So this is a luciferase reporter downstream of activation of SRF, and she finds that it's lower in soft compared to stiff at 24 and 48 hours. And that was associated with this nuclear localization, very modest though, nuclear localization of MRDIF on the stiffer substrates. So this is kind of consistent with the story that people have been reporting for cardiomyocytes, and we got the same story for YAP-TAZ, again very consistent with the idea being that these are direct actin-binding transcription factors that are involved in regulating fibrosis. But then Bailey did CRISPR-Cas9, she did siRNA, and she tried to do a knockdown. And this is showing you the knockdown of MRDF, and this is showing you the knockdown of YAP. And if these knockdowns were effective, this would not be 1. This should have been much higher, much lower in this case when you take away those cofactors. So she found that, in fact, these elements, SRF and TEAD, were definitely being driven by substrate stiffness, but that the YAP-TAZ and the MRDF were not involved in regulating that phenotype. And this is a scale of 0.25 to 1.75, which we don't consider significant. So this is showing you really nothing's happening if you take away YAP or TAZ. So I said, okay, well, there must be something else that we can do. And so she looked at, instead of just focusing on YAP and TAZ, she looked at a number of different small molecule inhibitors. And verteporfin was one that's supposed to promote elevated YAP-TAZ degradation. And that was quite successful in changing cell morphology. And importantly, this has been sort of the holy grail, and we're really kind of hopeful about this, in reproducing the vacuolated morphologies that we expect to see in the juvenile nucleus pulposus cell. So not only do we want to get a rounded cell, but we actually want that cell to have active membrane transport of internal membranes that will pump up these vacuoles and provide an inflation for that cell. So verteporfin, we're just learning about it and trying to understand its role as a small molecule inhibitor of fibrosis in these systems. And this is just going through, instead of working with laminin, we're working with peptides. We think peptides are a better way to go. We can, ideally, this is Marcos who's working with me now. Ideally, we want stiff substrates. So we want injectable, stiff, mechanically load-bearing signals, biomaterials, but we want them to induce the right cell phenotype. So by controlling peptide density, we can control both the cell morphology as well as the presence, the re-emergence of these vacuoles that we like to see in the cell type in the adult. So I took you through a lot in 2 really different stories, but all about the disc. But hopefully, the takeaway is that we do need to do basic science before we can go to translation and therapy, and in this case, both understanding pain and in understanding why cells die, we need to look at basic science and development. And we continue to be really interested in both cell-cell and cell matrix interactions and small molecule regulators, and we will be continuing to look for the best bioinstructive materials that we can use to manipulate cell phenotype. And that's what I hope we should continue to do as bioengineers. So I've given a shout out to the people in the group that have really contributed to most of this work. And I will stop there, acknowledge my funding sources and disclosures, and be really happy to take any questions.
So do we have any questions for our speaker?
Hi, Lori. Thank you. That was a really great, informative talk. I have a lot of questions, but I'll try to keep them to a couple. So first and foremost, I guess the question is, for example, with a cell therapy, if you inject a cell directly, like you mentioned several of these companies are doing, how long do those cells stay alive in the disc? And, what's the mechanism of sort of alleviating the pain? Do people know this?
Well, no, people don't really know. I think that most of the people involved in commercialization will tell you they don't want the cells to stay alive very long, right? Because it would be a problem in the long term if those cells were present for an extended period of time. They want them, just as if you think about Osiris and all the MSC therapies, they want to inject a healthy cell that's going to have a healthy Q and have the other cells take over, particularly in the case of allogeneic cells. There are some people who will say the host kills the donor cells in under 48 hours. There's some element of that. We were working here a little bit with iPSCs, a little bit mostly with primary autologous cells, delivering them in a carrier where they shouldn't leave to see if we could get them in there for a longer period of time.
I see. And so then, you said the Q, which is likely
Yes.
the key here. My question is, A, what is that Q? And B, do you need the Q, or can you just inject like a biomaterial that has the right mechanics that can essentially be like an artificial extracellular matrix to kind of plump up and be a hydrogel and plump up that area?
Right. So there's decades of work of people trying to inject just the biomaterials. And we haven't had, I would say, I guess I have to be careful here, but I would say we haven't really had a slam dunk with that at all, not even close, partially because the cells that are resident in the disc at that point are like macrophages and things, and they just kind of chew up anything that can be proteolytically degraded. And then people have worked with synthetics, obviously, for decades. Medical was one of the first devices that people have injected. Again, there are big problems with unless that biomaterial can be incorporated and be a well-received implant, then we are not optimistic that it's going to have a long-term outcome. And your question about, well, what's the magic factor? We're all, right? I wouldn't be here if I knew what the magic factor was.
Other questions?
So on the topic of the laminin stuff that you've been working on, have you done any work looking at the domains of the laminin that you think are the important part for that phenotype?
Yes, we have. So I think the cells largely bind to those LG1 to 5 domains, and my student Marcos has picked out the peptides that he's working with from integrin or non-integrin, some syndican-mediated binding domains in those LG1 to LG5 domains. And so I think we think that's where it's happening, but we haven't, I think if it works, we stop caring, but we probably need to pay more attention to that.
I'll take the last question if that's all right. I'm
Sure.
wondering about your companies that you mentioned.
Right.
And the cell sourcing for that. Could you talk a little bit about the cell sourcing and then how that's used in the intervertebral disc?
So there have been a host of companies that have developed either, and I think it's pretty widely used in the US and in Europe now, like where autologous cells are harvested from the defect site. So you might go in for cartilage repair. Cells will be harvested from that site, expanded, placed on a membrane, and then reimplanted back in that defect site. There are multiple companies that are doing that with autologous cells. And then there are companies that are working with allogeneic stem cell sources that have proprietary, their patent technology is on the marker expression for those stem cells. And mesoblast is certainly the most famous of those. But Osiris has also had a long track record in this country of success. And then those are allogeneic cells that are selected. They go through a bioactivity assay to show that they're doing what they think they're going to do, and then they inject them back in. They're very popular. I'm not aware of any insurer who is covering those, but they're very popular, and especially if you leave the country, you can get that procedure anywhere you want.
Do you recommend it?
Yeah. Oh, well, of course I don't recommend it because I, am a little bit more, concerned about what is actually happening in my own body. But lots of people have had, I can tell you lots of people have had allogeneic stem cells injected in their knee joints and shown that there is an anabolic effect that promotes a tissue regeneration, at least for a short period of time.
Cool. So I'm going to invite my Associate Director, David Otto, to come down, and we'd like to present you with something special.
Okay. Turn this up.
Thank you, everybody, for coming, and I'm going to ask if you would to please go out that side. We have another class that's coming in, I believe, on this side. So please go to that side, and if you have a desire for a sandwich on the way out, I think we have some more food. Thank you all for coming.
So during development, we think that they're really important because they are responsible for, like, they're very turgid. They're mechanically, we did like micropipette aspiration work early on, and they're much stiffer than other cell types in the region.